Just to review, the homework 6 is due to be submitted to Blackboard before class on Friday. And then today from 2 to 3:30 over in the Student Center is the State Department, who apparently are looking to hire construction and engineering related people. So if you're interested in learning more about that, it's over in the basement of the Student Center today from 2 to 3:30. And then the reminder about the Army Corps Spotlight Series lunch on November 9th. So we're talking about wastewater microbiology. This is chapter 22 in the book, and there's really no problem solving that goes along with this. It's just kind of informational. But this content, even though it's not problem solving, still is subject for inclusion on the final exam, and we're going to have at least one more quiz before the semester's over as well. So it's likely that I'll throw in some sort of a microbiology related question on one or both of those. Any announcement questions before we go? All right. We're just going to talk about different categories of microorganisms today, in part so that we can learn a little bit about the different roles that they play in wastewater treatment. And the first type of microorganism that we'll talk about is those which don't have a centralized nucleus, prokaryotes. They're relatively small, and oftentimes they have the capacity for movement with the flagellum. The purpose of this flagellum is to move them around in the wastewater, and what they would do is move towards a source of food. So if in a wastewater treatment plant there is like a concentrated gradient of BOD, then they would use the flagellum to move towards that. And because they're relatively small, prokaryotes are kind of thought of as being the prey. A lot of the figures that are presented in your book, there are different figures that show, depending on the residence time of cells in a wastewater treatment plant, different dominance of predators versus prey. And so when you look at those figures and you're wondering what's the predator and what's the prey, prokaryotes are the prey. They're at the bottom of the food chain. Because they're small, they don't feast on suspended waste. They are going to be taking up dissolved waste. And so after primary treatment, there still remains some suspended BOD and some dissolved BOD. And the prokaryotes will be taking up the dissolved BOD. Whereas eukaryotes, because they're larger in size, have the ability to consume flocculated particles, and in fact they can also prey on the prokaryotes as well. Jacob, any questions? No? All right, so I'll point out that the motility, the ability of the eukaryote to move around in the solution is a structure similar to the flagellum. It's just called the cilium. And eukaryotic bacteria aren't the only things that have cilium. They're also present on larger organisms as well. So that's an important feature of these bacteria, is whether they can move around on their own or whether they have to be suspended through aeration of the aeration basin.
So there are cells with one cilium?
Yeah.
I thought, okay.
Yeah.
In biology class I always thought it was a bunch of cilia around the cell.
We're going to look at a video today that has, it's kind of like an X-ray chromatography of some different microbes that are in wastewater, and I think one of the slides that they show is one that just has one dominant cilium, and there may be others around it that are used to move the liquid around the surface of the cell, because the cilium, as I understand it, serves two purposes, and one is to provide movement of the cell, and then the other is to move fluid around the cell. And so I think if we see long ones and small ones, they may be forming or serving different purposes. Bacteria are mostly water, but this table that we have on the screen here is the dry weight. And so if you set aside the water, which is going to be, upwards of 90% or even more of cells, then the major source of the mass that remains is protein and nucleic acid. And then if you break it down by the cell elements, you can see that a large part of the protein is carbon. And so this is important because for bacteria to grow, they need sources of carbon and they need sources of energy. And we're going to be talking about different ways that they get their carbon and different ways that they get their energy based on different categories of pathogens and microbes. So these two things that are required, energy and carbon, are the two main constituents of what they're in the treatment plant looking for. And then there are a few trace elements that are really important as well. And the ones that are typically limiting are nitrogen and phosphorus. The other elements like sulfur, potassium, calcium, magnesium, they're all required, but they're typically in much larger abundance, and so they're not the limiting reagents for the growth of new microbes. So a general model for this is that the microbes are taking the organic material, and some of the slides that we'll look at today, sugar is just used as a general substitute for some sort of a organic substrate. So it's not always sugar that's the BOD. Sometimes it's longer chain materials that are more complex carbohydrates than a simple sugar. But there has to be some sort of a nitrogen source, a phosphate where it's getting phosphorus from, and then oxygen, and it's going to form new cells. Nitrogen can be problematic in the environment. If it's going to stimulate bacterial growth in a wastewater treatment plant, it's also going to stimulate bacterial growth in the environment. So as much as possible, we want to remove not just BOD, but we also want to remove nutrients from wastewater, because it could, in theory, be possible to remove the BOD but leave the dissolved nutrients in the water, and then we're discharging the water into a stream or into the ocean, and there's not the same negative effect as if the BOD was there, but having the nutrients can still just maybe kick the can down the road, and there could be stimulated growth later on. And so we're looking for ways that we can not only break down the organic material with the microbes, but through different sequencing, we can play tricks on the microorganisms and make them think that they're going through periods of starvation in which they take up additional nutrients beyond what they normally would. And so there's ways to cycle them between aerobic and anaerobic conditions, and by doing that, the microorganisms will take up 10 times, 15 times as much of the nutrients that they ordinarily would. So we'll talk about that nitrification, denitrification process a little bit later, but I wanted to mention it here because the inorganic elements that we're trying to move, nitrogen and phosphorus, are particularly insidious when they get into the environment. So autotrophs get their carbon from inorganic sources, specifically carbon dioxide, which is dissolved in the atmosphere. It's one of the gases in the atmosphere, and it also gets into solution. So that's where autotrophs get their carbon. So you and I are obviously not autotrophs. We don't get our carbon from carbon dioxide, although we generate carbon dioxide through our respiration. We're not using it as a carbon source. We get our carbon from organic material like hamburgers and potato chips and BOD. No, not BOD. But the heterotrophs that are in wastewater do. They get it from dissolved and suspended BOD. They're organic material. So likewise, autotrophs and heterotrophs get their energy from different sources as well. Autotrophs, the ones that are using carbon dioxide as their carbon source, get their energy either from photosynthesis or from oxidation reactions utilizing ammonia. So one autotroph, nitrobacter, is used in wastewater treatment to remove nitrogen from dissolved wastewater, whereas heterotrophs are getting their energy source from the oxidation of organic material like sugars and carbohydrates and starches. But there's also a lower energy process. The fermentation of organic matter is in the absence of oxygen. They can gain energy from organic matter, and it's much slower and it yields less energy, and so you have to provide much longer exposure times to break down waste if it's being done through a fermentation process. But the heterotrophs that we will have growing in an aeration basin for activated sludge move very quickly, and then typically we'll use fermentation during sludge treatment. The sludge that we suck off the bottom of the secondary clarifier can be broken down over a month, 2 months of contact time in a digester, and through the process of fermentation it actually can form methane that can be used to heat water inside the wastewater treatment plant. So it's generating enough energy from the anoxic breakdown of organic material that you can heat up and speed the reaction of the sludge digestion. So if we look at a schematic model of this, the aerobic heterotrophic bacteria are taking in organic compounds like sugars and carbohydrates. They have nutrients like phosphorus and nitrogen. They're also utilizing oxygen as an electron acceptor, and from that they're generating carbon dioxide, water, and then new cells, which, because of cell division, is going to increase the speed of the utilization of BOD as long as we are in the range of having enough cells that we can support their growth. Now, aerobic autotrophic cells are going to be using carbon dioxide rather than organic materials, and instead of just forming carbon dioxide as the output, they're forming nitrite and water. And then finally, the anaerobic materials are also breaking down organic compounds, but they're doing that in the absence of oxygen, and they form methane as the output. And so, there's not enough energy being recovered there that you could power a city, but there is enough energy there that you can warm up the sludge that's being treated, and then the process is a little bit faster than it otherwise would be. So oxygen used aerobically generates a lot of energy. This chemical reaction of glucose and sugar will generate 686,000 calories per mole of heat energy, whereas if you're breaking down glucose anaerobically, it forms lactate and much less energy. And so the reaction is slower, and so per unit of BOD removed, the aerobic process is a lot quicker, but it's just providing the oxygen is electricity intensive, because for the bacteria to consume the oxygen, we usually have to provide it through pumps, although there are other ways of providing the oxygen. If you have a very large lagoon, then the oxygen can just be provided by the action of the wind or by diffusion of the air through the water-air interface. But you need quite a lot of surface area if you're just using a sludge lagoon, rather than like in urban areas where space is limited, you have to concentrate the delivery of oxygen through intentional aeration. A secondary way of providing oxygen that doesn't require electricity, besides just a big lagoon where it's diffusing, is with algae. So, in a lagoon, if there's algae growing, then that can also be a source of oxygen getting into the water, independent of having to consume electricity to do it. Now, sometimes people use the phrase anoxic and anaerobic interchangeably, but technically those two terms mean slightly different things. In anoxic conditions, there's no free oxygen, that is, there's no dissolved oxygen, but there are oxidized compounds like nitrate present. And so the type of chemical reactions that can happen in an anoxic chamber are different from if it's fully anaerobic. If it's fully anaerobic, then that means that there's no oxidized compounds present as well. So this is just in this table illustrating that you still have in an anoxic environment nitrate present as an electron acceptor, but that's not the case in an anaerobic environment. In an anaerobic environment, the electron acceptor will be an organic compounds or carbon dioxide, and so that's when methane is going to be formed, is in a truly anaerobic environment. So you can see what this is illustrating is if water is going in a circulating pattern and the wastewater is only being aerated in a certain part of this circulating chamber, then if the water is moving in a clockwise fashion, and here's our aerators, then the air would be consumed as it's moving plug flow. The water's moving clockwise. The oxygen would be consumed, and then there's going to be a certain part of this circular racetrack configuration when all of the oxygen is used up, but we still have the nitrate present, and so different bacteria would grow in the left side of the reactor compared to the right-hand side of the reactor. And as different bacteria are growing, they're going to have the ability to, for example, larger bacteria will consume flocculated groups of bacteria, and a variety of different microorganisms in a wastewater is an indicator that you have a stable population that is healthy and that it's not going to be wiped out by variations of variations of BOD or the presence of low concentrations of toxic materials. If you have a broad diversity of a population in a wastewater treatment plant, then it's more resistant to the variations in day-to-day composition of the waste. And then the other advantage of having this part of the reactor being anoxic, part of it being aerated, is that this is the kind of configuration that allows you to remove nitrogen. And so instead of discharging ammonia to the receiving body or dissolved nitrogen, it turns into nitrogen gas, which just gets into the atmosphere and it isn't considered a pollutant whatsoever. So we'll talk more about how this kind of cycling approach can be used to reduce dissolved nutrients. Bacteria are single-celled, and they're the primary feeders or the prey in activated sludge, and they have a variety of different shapes, and the shapes sometimes influence their settling properties and the types of flocks that they're able to form. So there are some that are spherical, some that are cylindrical, some that are helical in shape. In terms of their optimal growth, we want the pH to be relatively neutral because they're so small and their cell walls are relatively thin that they're not resistant to acidic or basic environments. And there are different types of bacteria that prefer cooler environments, some that can adapt to moderate temperatures, and some that thrive under really warm conditions. And so, another thing about this idea that having a wide variety of bacteria in an aeration basin is that if you have some that are able to handle cooler temperatures, then as the seasons change and as the wastewater temperatures shift, then they continue to take up BOD in an effective way. So it's just another advantage of having population diversity in your wastewater treatment plant, is that the seasonal temperature shifts can be tolerated. The ideal ratio for BOD removal and nutrients is a mass ratio of 150 grams of BOD for every 4.7 grams of nitrogen and every 1 gram of phosphorus. And so this is what's needed for the growth of new bacterial cells. And so ordinarily domestic sewage is going to have more nitrogen and phosphorus. And so just the ordinary growth of new cells in the aeration basin could use up all of the BOD but still have leftover nutrients. And that's why we have to get a little bit creative with things like cycling the oxygen on and off in an aeration basin to trick the microorganisms into taking up more than 4.7 grams of nutrients for every 4.7 grams of nitrogen for every 150 grams of BOD. So just to put it all together, this table summarizes the different types of bacteria, the types of reaction that they're going to be accomplishing, what is their source of carbon, their source of energy, and then what's the output product of the growth of those bacteria. So it's not common that we'll use iron reducing or sulfur reducing bacteria because those constituents are pretty small on a mass basis of what's in the wastewater. But methanogenesis and acid fermentation could be pretty helpful in sludge digestion. And then of course the aerobic heterotrophic bacteria are kind of our bread and butter, so to speak, of breaking down the BOD in the wastewater reaction. Fungi are a problem because they don't do much for us in taking up BOD relative to the problems that they can cause with correct settling of sludge. They tolerate a much wider range of pH than bacteria do, and so they can be persistent, and filamentous fungi, filamentous just means that they are long and stringy and that they kind of clump together, and because of that they settle poorly. Remember that spherical shapes settle well because you have a relatively large mass and a low surface area. But if it's long and filamented, then you're going to have a relatively large surface area relative to the mass of it, and so it's going to settle more slowly. And it's not just that it settles slowly, but also if you have a bunch of interlocking fungi in a wastewater treatment plant that is generating nitrogen, then it can actually cause the sludge to float to the surface rather than to settle down like it's supposed to. So we don't like fungi in the wastewater treatment plant. Algae, I guess you could say we're indifferent about algae depending on where they are. In a sewage lagoon, algae are beneficial because they can provide oxygen, which speeds up the breakdown of BOD compared to if it's just going to be oxygen that's supplied by diffusion. But there's different types of algae that can put off toxic substances. Like in the case of blue-green algae, there have been a couple of blooms in the Ohio River over the past decades where blue-green algae will take hold, and they have a toxic substance that they let off, and it kills fish, and it can actually even be dangerous for people and the wastewater treatment, the drinking water aspect of it. So we don't really have to worry about blue-green algae in wastewater treatment plant, but it's just something to be aware of. There can be some algae that are just diatoms, are really small individual cells of algae that aren't the long green kind of grassy looking algae that you're maybe familiar with. Diatomaceous algae are kind of much smaller. But as I've already mentioned, the usefulness of algae is that they can provide oxygen in sludge lagoons. They also will take up appreciable amounts of nitrogen and phosphorus as well. So here's a couple of slides that just shows the wide variety of shapes and appearance of algae that you could conceivably see in a wastewater treatment plant. Now, protozoa are secondary feeders, meaning that they will be feeding on the eukaryote, and that they are considered an indicator that you have kind of a mature sludge that is well established. If there are feeders, then that's the predator that's breaking down some of the prey. And there are different categories of protozoa, amoeboid and flagellated, but the ciliated protozoa are the ones that are the most dominant in wastewater treatment plant. And here's that video I was mentioning earlier that illustrates what they look like. Mute this. So it's kind of interesting to see them moving about. How big? Well, let's see. They were saying the magnification here, 250 to 800 times. Yeah, I don't know. They're pretty small. Okay, this has got an idea of some sizes. 100 microns, in the range of 100 microns. Rotifers are even larger, and you can see here that they are thought of as consumers of the flocculated bacteria and particulate organic matter. And so rotifers are another indicator that you have a diverse population of organisms in a secondary treatment. Now, I've mostly been talking about aeration basins in secondary treatment. That's called a suspended growth model, where the bacteria are floating around in the wastewater, and by the way that we're getting a high concentration of bacteria in the wastewater is by we're collecting sludge at the bottom of the secondary clarifier and re-injecting it into the aeration basin. So that's suspended growth. But there's also fixed growth, where instead of having the bacteria floating around in a tank, we provide an environment for the bacteria to attach itself to either rock or plastic pellets or some sort of a fixed media, and then we spray wastewater over that media, and so it's sometimes called a trickling filter. And we'll talk about both suspended growth and fixed growth, but these bacteria can be present regardless of whether it's a suspended growth or fixed growth. They can be in the water that's being treated, they can be living in the boundary layer that's on the thin film on the media. There's usually like an organic slime that'll set up on the rocks or the plastic pellets that are the media for fixed growth. All right, so we're going to finish up today's class by watching this video. Let me pause this here. They could infer from that, the study of all the different animals, how the population of zebras and how the population of like the holistic health of the ecosystem. And if they knew that there was too many hyenas but not enough cheetahs, then they maybe from that could tell that one particular prey was too low. And so what they've done is through regression analysis, they've correlated different types of indicator species with the potential cause for that. Now, the book used a couple of abbreviations that we'll get into a lot more detail later, but one of them was the idea of F to M ratio. There's only a handful of things that an engineer can design and control in wastewater treatment. like you don't necessarily have control over the BOD itself. That's just a byproduct of whatever the waste that's coming into the treatment plant is. But F to M is the food to microorganisms ratio. And so the food is generally fixed. That's the BOD that the microorganisms are going to be consuming. But the microorganisms mass is what you can control through recirculating the settled sludge that you get at the secondary clarifier. So that's one of the important parameters that was mentioned in that video, where if you have certain kinds of indicator organisms, it can mean that the F to M ratio is too high versus too low. One of the other parameters it was mentioning that we can control is the dissolved oxygen. And the dissolved oxygen is added to the aeration basin through fans and blowers, and it's very expensive to provide oxygen to the microorganisms, and so you kind of want to chintz out on that as much as you can, be sparing and judicious in the amount of electricity that's used, and so you want to provide just enough to support the bacteria, and so that they're removing the BOD quickly, but every additional gram of dissolved oxygen that's being provided costs money to get in there. The other thing that was mentioned is MLSS, mixed liquor suspended solids. And that's the concentration of bacteria that are in the aeration basin. And so it's another parameter related to the F to M ratio, which is, what concentration of bacteria do you want in the aeration basin? In a given liter of water, how many grams of activated sludge should there be in order to break down the waste? And then the one other parameter that it didn't talk about, but it kind of hinted at indirectly. Let me go back to the slide or the part. Let me just mute this. See if I can find it, because there was a chart. This one. All right. What they're not showing on the horizontal axis is how long the cells stay in this cycle of going between the aeration basin and the clarifier. And so it's the mean cell residence time. Remember that we've said that there's two different residence time. There's the hydraulic residence time, and then there's the residence time of the cells. Because in secondary treatment, let me go to a diagram that's illustrating secondary treatment. In secondary treatment, the sludge that settles in the secondary clarifier, some of it will be sent for digestion, but some of it is recirculated into the aeration basin. So the question is, what fraction of this sludge gets wasted, and what fraction of this sludge gets reintroduced into the aeration basin? And the relative balance between how much is wasted and how much is reintroduced into the aeration basin determines how long the average cell stays in this loop before it's discharged. And so if cells stay in the loop for a relatively short period of time, then you may not have the opportunity to see a good variety of organisms set up. So this would be characteristic of a very short mean cell residence time, whereas if you have a very long mean cell residence time, you'd have maybe an overabundance of the secondary predators. And so, the balance between eukaryotes, prokaryotes, rotifers, and so on, typically microorganism populations that are somewhere in the middle are the most productive in removing BOD quickly and the most resistant to variations in operating changes. And so another thing that these population studies can tell you is kind of if you're achieving that right balance of wasting sludge versus recirculating sludge. All right. Well, that's all the time we have for today. Remember that by Friday you need to finish working on homework number 6. And if you have any questions about that, feel free to stop by during my office hours or send me an email. I'll see you on Friday.